All right. Hi, everybody. So we're going to go through a quick case study involving signal transduction pathways. And we're going to talk about specifically an example using the fight or flight response. So the question that I have for us to get started is what causes the experience of the fight or flight response in mammals? And we're going to look at the mechanisms and the signal transduction pathways that go along with this. Now, if you're following along, there's an option for the handout. So if you look at this handout, it'll be attached in the video notes, as well as if you're in my class, then there'll be a printed version of this for you, but you can follow along using this signal transduction lecture guide, and that'll be helpful for you. So when we're looking at the fight or flight response, there are some vocabulary terms that we need to be aware of before we dive into the nitty-gritty details of signal transduction pathways in the fight or flight response. So the first important vocabulary term that we need to know is a relay protein. A relay protein transmits a signal from a receptor protein and a catalytic enzyme. So remember, catalysis means that it is going to perform a chemical reaction. So these proteins are going to be involved in some sort of chemical reaction. They're usually fairly large. These relay proteins are fairly large. They require some sort of activation, and they do not diffuse through the cell quickly. Okay, so relay proteins, a good example of a relay protein, is a G protein. So if we're looking at this example here, this is a G protein coupled receptor. This is a G protein, and then this is the enzyme that the G protein is eventually going to activate. So this is just the setup. This first frame here is the setup. And if we go to this diagram, this top right or top left, frame is the same frame. So if we look at this example, I'm just going to go through this example pretty quickly. We have a G protein that is eventually going to be activated by the presence of GTP. So if we look over in the second frame, it's going to, and I'm going to move my little screen here, it's going to be activated by the presence of GTP. Now, GTP is not going to replace GDP until the ligand, the signaling molecule, binds to the PCR, the G protein coupled receptor. Okay. So the sequence of events for that is signaling molecule or ligand binds to the receptor. The receptor causes an attraction, it changes shape, and that's going to attract the G protein, which then causes the G protein to attract GTP. And then in this third frame, we see here that the G protein has been activated, and GTP is part of that activation process. And it's going to move over to the enzyme that's needing activation. And that is what's going to induce the cellular response. After this process occurs, the whole system resets. GDP reassociates with the G protein, and the receptor molecule returns to its original state. Okay, so this is the sort of essential function of a relay protein, and it does exactly what it sounds like. If you've ever seen a relay race, it's the transferring of a signal from one protein to another protein, the same way that you hand a baton in a relay race. Okay. So this is what that process looks like. This is just an animated version. Here we have our signaling molecule, which is epinephrine. The surface receptor, the G protein is activated. It's then going to activate this molecule, adenylyl cyclase. And adenylyl cyclase is going to turn ATP into cyclic AMP. Now, this diagram is not showing GDP being replaced by GTP. So in this space here, GDP is going to be replaced by GTP. So it's important to remember that this diagram isn't necessarily showing all of the pieces. Another important signal transduction pathway vocabulary, and that is a kinase. There's a typo here that says kinsases. It should say kinases. A kinase is an enzyme that phosphorylates or adds a phosphate group to another molecule in order to activate that molecule. Typically, they are going to activate other proteins. And this activation, this phosphorylation, induces a conformational change in the protein's structure. So if we look at this specific example, we have protein A, just generic protein, and a kinase is going to be an enzyme that takes ATP, the energy involved in ATP, and it's going to transfer that energy to this protein, and then it becomes phosphorylated. Now, this is also showing the potential reverse reaction. So you can dephosphorylate this protein using phosphatase, which is another type of enzyme, right? So kinases are going to be really important when it comes to transferring energy and activating other proteins. So let's do an example. We have an example here using the G proteins as well as the G protein coupled receptors. And in this example, they have divided up the G protein into its component pieces. So G proteins are made of an alpha, a beta, and a gamma subunit. So that's what we're seeing in this inactive state. In the inactive state, GDP is going to be bound to the alpha subunit. Once the ligand binds to the receptor protein, GTP is going to replace GDP, and that is going to induce the downstream molecules. Downstream effectors just mean that they are part of transduction, and they are going to induce the rest of this signal transduction pathway. So my question for you here is, would a G protein be considered a kinase? Why or why not? I want you to pause the video for a second and try to answer this question on your own, and then I'll give you the answer when you come back. So hopefully you've paused the video at this point. When you're looking at this example, would G proteins be considered a kinase? And the answer is no. Even though the G proteins are involved with GTP being replacing GDP, they are not going to be considered a kinase because the G protein itself is not the thing doing the phosphorylating, right? If we go back to a kinase, the kinase is going to be the molecule that is phosphorylating something else. But in this case, the G protein is not phosphorylating anything, okay? So let's look at another example. So we're looking here, and you guys probably recognize this if you have done the signal transduction POGIL activity, POGIL activity. So based on this image, what is the benefit of having relay proteins as opposed to direct receptor response pathways? So instead of the receptor immediately inducing a response, why do we have this in-between step of transduction? Take a second to pause and think about this question. Hopefully you saw my hint in this question. The hint here is amplification. So one of the benefits to having relay proteins is that you get amplification. So we can see in this example that protein 1, relay protein number 1, is going to influence and activate multiple pathways using protein number 2, relay protein number 2. And what this does is it amplifies a signal so that one ligand can produce a broad range of responses. So it can produce a lot of responses as well as potentially other types of responses. So for example, there's an image here that I'm actually just going to bring up for us because it's going to be relevant for this discussion. So when we're looking at this diagram, again, one of the main ways that relay proteins are beneficial for signal transduction pathways is that they amplify a signal. They make the signal bigger, so you get more of a response. So a great example of this is the chemical receptors in your nose. So for example, you have hundreds of thousands of different chemical receptors in your nose that are going to trigger certain olfactory senses in your brain. But oftentimes when we smell something, we're only smelling tiny, tiny, tiny concentrations of that particular substance. So what the signal transduction pathway does in that case is it amplifies the signal so that we actually can identify the smell, and it'll send enough signals to our brain. And that's how we've evolved to be warned of, things like poison or rotten food. All of those sensations are a result of signal transduction pathways and amplification via signal transduction pathways. So I wanted to show you this diagram. You're going to see this diagram later as well, but this diagram is showing the possible mechanisms of several signal transduction pathways. So if we look up here at the G protein coupled receptor, the G protein is not just involved in one pathway. It's involved in a variety of different pathways. And this is very common in relay proteins and signal transduction pathways. One protein typically is going to be involved in a variety of different pathways. And I encourage you to potentially pause the video on this diagram, because there's some interesting names for these proteins that I encourage you to try to find. All right, so continuing our discussion of important vocabulary, let's take a look at this diagram. So what is the relay protein in this signal transduction pathway? What is the second messenger? And what is the primary messenger in this pathway? Go ahead and pause the video, and then I'll come back to you when you get back. Okay, hopefully pause the video. We're looking here. What is the relay protein in this signal transduction pathway? Well, that's going to be the G protein here. What is the second messenger? Well, luckily it's labeled for you pretty clearly. That's going to be CAMP, cyclic AMP, adenosine monophosphate. So what cyclic AMP is actually ATP with a cyclic molecule attached to the ATP, and then two phosphates are removed. So it becomes monophosphate instead of triphosphate. So that's the second messenger. And then what is the primary messenger in this pathway? Well, the primary messenger is going to be epinephrine. Epinephrine is going to be the initiant of this pathway. And we're going to come back to this diagram in a moment when we start talking a little bit more about the fight or flight response. Okay, so we've covered a little bit about first messengers and second messengers. First messengers are going to be the ligand. Second messengers are going to be small, typically non-protein molecules or ions that can spread really easily throughout the cell and transfer a signal much more quickly. So in this example, cyclic AMP, it's not just one cyclic AMP that's going to be made. It's a variety and a multitude of cyclic AMPs that are going to be synthesized. And that's going to cause a rapid succession of messengers or, of pathways triggered throughout this cell. So cyclic AMP is an example of those second messengers. And we see that here in this diagram. So this is, again, epinephrine with a cell surface receptor. It's the same diagram that we just saw on the previous slide. G protein is going to be activated. It's going to cause adenylyl cyclase to convert ATP into cyclic AMP, but it's going to do this in rapid succession. So one signal molecule, in this case epinephrine, can trigger the production of multiple cyclic AMPs, which can be beneficial when you want to amplify a signal. Additionally, we want to talk about something called a phosphorylation cascade. So a phosphorylation cascade functions in two main ways. Again, amplification is a big one. You want to be able to amplify a signal with a very, very small concentration of your ligand, right? So amplification is a big one. In this case, the kinases are going to stay active after catalyzing multiple reactions. And so you get these kinases that are phosphorylating their enzymes, they're phosphorylating their products, and that's going to happen multiple times. So a single kinase can activate multiple of their products, and so that's going to cause rapid amplification of any particular signal. Another function of phosphorylation cascades is crosstalk between different pathways. So if we go back to this diagram, When you use a phosphorylation cascade, that can create a domino effect, which triggers multiple pathways. So for example, if you have a phosphorylation cascade between RAF and MEK, that can potentially cause a rapid increase in the rest of this pathway, which can eventually lead to greater gene regulation. So again, big concepts in signal transduction pathways are amplification of a signal and then crosstalk between different pathways. So let's take a look at an example. Let's say that we want to go through the process of mitosis, which is actually what's in the end of Unit 4 for AP Bio. In the end of this unit, we talk about the process of producing new cells. Now, hopefully you've seen this before in your general biology classes, but this is just a review. The process of creating new cells is where we multiply the DNA, we replicate the DNA, so we have 2 copies of DNA, then those copies of DNA get pulled into separate cells. So at the end of this process, you get 2 complete, fully functional cells. So I want you to pause and think about, what are some of the reasons we might need new cells? Hopefully you've had a chance to think about it. I want you to think about, what are some of the reasons we might need new cells? Well, some of the reasons are when cells get damaged. Your cells are getting damaged all the time. Your skin cells die, they are shed off. When you look and you see those nice little particulates in a sunbeam, those are usually a lot of dead skin cells. And so our body needs to replace those skin cells. Another fairly obvious example is growth. We do not start out as fully formed adults. We start out as children. And in order to go from tiny babies to fully formed mature adults, you have to make more cells in order to get to that size. And that's this process of mitosis. Old cells die, new cells are going to be needed. We need to be able to replicate those new cells. So here is an example of a signal transduction pathway in which the ultimate goal is cell growth and division. So we're trying to go through the process of mitosis. We're trying to trigger mitosis within this particular cell. Now, what's going to happen in this particular example is a growth factor is going to be received as the cell signal. So growth factor is going to activate the phosphorylation cascade. And in this case, it's going to trigger 2 responses. It's going to trigger a cytoplasmic response as well as a nuclear response. In the cytoplasm, it's going to start to increase the translation of certain mRNAs, and that's going to produce certain proteins that trigger cell division. But you're also going to get the production via the nucleus of additional mRNAs. And those mRNAs are going to go on to produce additional proteins. So through this phosphorylation cascade, you get this domino effect, this waterfall effect that dumps this massive response and transduction mechanism into the cell to induce cell growth and division. And we're actually going to come back to this example later on when we talk about cell division. So let's think about an example. What situations might we want to stop cell growth? What situations might we want to stop cell growth? And how might we go about this? I'm going to give you a second and let you pause the video. So some situations where we might want cell growth to stop are when we've had damaged cells that have been replaced, but we don't want to make any additional cells. If we continue to make additional cells on top of the ones that have been damaged, that is when we end up with a tumor. A tumor is a cell that never got the signal to stop dividing. So how might we go about this? Well, we can stop a cell from dividing by inhibiting growth factors. We can stop a cell from dividing by inhibiting these proteins, so MNK1 or CMYC. Those are all proteins that we could potentially inhibit, but the best way is going to be to inhibit or somehow change the growth factor, because that's going to stop this whole process from happening. What might be the benefit of having simultaneous gene regulation and cytoplasmic response to a stimulus? Pause the video and take a second to think about it. Okay. What do you think? What might be the benefit of having both gene regulation, so transcribing new genes, as well as a cytoplasmic increase in the translation mechanisms? Well, again, it comes down to amplification. You get this process where the proteins that make that cause cell division, you need the RNA to make those proteins. And so you get the proteins, but you also get the RNA. And so you get this push immediately into the process of cell division and growth. So again, you get a faster response, you get a more controlled and specific response as well. And you also get amplification of that response. So when I say more controlled and specific, you get a really directed response, right? When you are specifically controlling the mRNAs that are being released out of the nucleus and into the cytoplasm, if you have a really high concentration of a certain type of mRNA, that means the ribosomes are likely going to translate that specific mRNA because there's so much of it present in the cytoplasm. So I think I'm going to stop this video here, and I will film the rest of the next part, and then we will see each other soon. Thank you.